Exercise 22 deals with the digestive physiology, and there is an accompanying file that goes along with this that does show pictures of some samples of each of these tests. What this experiment and exercise deals with is testing for some of the main macromolecules for nutrition, your carbohydrates, your lipids, and your proteins. And with the carbohydrates, we have two different tests that we run. One is looking for simple carbohydrates such as glucose, these simple sugars, and another test is looking for more complex, specifically starch. So for the simple carbohydrates, what we do with this, we have samples, and like I say, you will see pictures of these in the file that goes along with this PowerPoint. All of that is on Blackboard. But to run the test for simple carbohydrates, you would have your sample. You have what's known as Benedict's reagent. It's kind of a bluish color reagent. And you add about 15, you have about a ml of the sample. And then you add about 15 drops to the sample. Then you need to heat it for 5 minutes. Now, some places will tell you you need to heat it up to boiling. You really only need to go to about 80 degrees Celsius. That's usually sufficient. And just leave it sit. Now, it's not critical that it has to be there exactly 5 minutes. If you get doing something and, oh my goodness, it's 7 minutes, I left it 2 minutes longer, that's no big deal. It's fine. It'll be okay. What you are looking for is a color change. As I said, the Benedict solution by itself has a bluish color to it. So the way you interpret the results, if it is still blue, that means there is no glucose present. If it's green, it means you have a small amount, just a very small amount of glucose. If it's yellow, there's a low amount. If it's orange, it's moderate. And if it's red, it means you have a very high amount of glucose in this. Now, to test for complex carbohydrates, like starch specifically, that's what we're doing. Here, what you do is, in your samples, once again, in test tubes, usually about a ml per tube or per sample. What you do is you just need to add about 2 drops of iodine. Now, iodine normally is kind of a yellowish brown color. You just add those drops to each of your sample tubes. If it stays a yellowish brown, that means there's no starch present. If it turns a very dark blue, almost black, some people will say it's like a dark, dark purple, but it's a very, very dark color, then that means it's positive starch is present. So it's either there or it's not. And for this one, you see the results right away. You don't have to heat it up or anything. You just room temperature, add the drops of iodine, and immediately look. If the color is going to change, it will happen right away. One thing I will say for testing for the complex carbohydrates for the starch, sometimes starch, if there's a high concentration, it kind of precipitates out of the solution. So make sure you mix the tube up well to get that starch in solution. To test for lipids, this is very easy to do. When we're doing this in a lab, we usually take some brown paper, like a brown lunch bag, and we just rip up some of that paper and then have the students put a drop of each sample on the brown paper. And then you just set it aside and let it dry. You guys have all seen this in various aspects. After it has dried, if there's a stain there, that means there is lipids. Remember, lipids are fats. If there's no translucent stain there, then there are no lipids in the sample. And like I say, you guys have all seen this. How many times have you been really hungry and you have French fries and you put them on a napkin, and then once you're done with eating the French fries, you kind of look down and there's a stain there? That's because of the fats from the oil that was used for frying the French fries. Or a pizza box. We've all gotten pizza. And the pizza box afterwards, you look at it and it's like, oh my goodness, it was kind of greasy. Well, what is grease? It's lipids, it's fats. So once again, this is a very easy, quick way of testing for lipids. And then the last test that we do, so it's the carbohydrates are both simple and complex. lipids, and then the fourth thing we test for proteins. For this one, you take your samples, once again, about 1 ml per tube. You have what's known as a biuret agent. This is also kind of a bluish color. You add about 5 drops per sample. And right away, you, once again, don't have to heat it up. You right away look for the color change. If it stays blue, that means there's no protein. If it turns purple, then there is protein present. So once again, this is telling you how to do these tests, and it goes along with the file that has actual pictures that were taken from running these experiments.